Welcome to the IB Biology IA Research Design walkthrough. So the research design is the first section and it's worth a maximum of 6 marks out of the 24. This first slide here just goes through the overview of the IA, read it if you need to. And then we've just got some extra information about the research design, and we've also got the marking criteria for research design. So just be aware that it's out of 6 and the marker will decide which band you fall into and then whether you deserve 3 or 4 marks in the second band or 5 or 6 marks in the third band. Okay, after you have outlined your research question, and that's a different video if you want to go and find it, the background information should be about a page of writing, and it's going to give context for the investigation. So I've outlined what, if I was writing it, what I would put in each paragraph. So the first paragraph would be kind of why you chose the project, what's the interest. In the old spec, you used to get 2 marks for this. You don't get 2 marks now, so this doesn't have to be in a huge amount of detail, but just kind of why you've chosen it, what's the reasoning behind choosing it. And it just kind of adds personal interest to the project as well. Second paragraph should be all about the independent variable. So you need to give as much scientific, like IB level scientific detail about your independent variable. So if your independent variable is temperature, for example, in an enzyme investigation, you're then going to explain about how temperature will break bonds within an enzyme and cause the active site to be a different shape, so therefore it will no longer be complementary to the substrate, so fewer enzyme substrate complexes are produced, etc., etc. So we're going kind of super scientific here. The dependent variable, the same, needs to be IB level, beyond if you so wish. At this point, you need to kind of have studies, so you want some other scientists who've done similar studies. Now, it doesn't have to be exactly the same study. It could be a study with the same independent variable, the same dependent variable. The whole idea about the IA is that nobody has done this project before, so you will not find the same study. And if you do, why are you doing it? Because somebody else already has the results. The background information needs to be fully referenced using Harvard referencing. You'll submit your work on ManageBack and it will go through an AI checker, so we'll know if you have copied and pasted off the internet or if you've used AI. My advice is don't use AI because A, we know, and B, the information isn't always as targeted as you need it to be. So by all means, you use AI sensibly for research purposes, but you need to reference everything. It's difficult to reference any sort of AI generator. So my advice would be not to use it. And there's a little example there of some background information. Okay, you're going to outline your independent variable. You need to state the unit and the range and kind of explain why you chose that range. So if it's temperature and enzymes investigation, you are choosing 20, 30, 40, 50, 60 degrees because you think that 40 is around about the optimum temperature, and therefore you will be able to see the slower rate of reaction underneath below 40, and you will be able to see when the enzyme is denaturing above 40. So just kind of explain why. The dependent variable, you need to state the units and say how you're going to take the measurements. So if you are using a gas syringe to collect a gas, then say that rather than, because you could be counting bubbles of gas. So you just need to kind of explain. If you want to put a diagram or a picture in there, then by all means do. And then control variables, you need at least 5 of them, and I prefer them in a table like this. So temperature as a control variable, obviously I've talked about that as an independent variable. It can't be an independent and a control variable. So if temperature was a control variable, you then need to say why it would be controlled. So I've given the example there that the temperature has to remain the same because if it increases, it increases kinetic energy, increases the speed that molecules move, and therefore the chance of successful collision, so that would increase the rate of reaction. So I've gone scientific. So you can't just say it needs to be controlled to make it a fair test or to make it reliable, to make it accurate, because these are the types of terminology that people throw around and they don't really know exactly what reliable and accurate means. So you're explaining scientifically here why we need it to be controlled. And then how you'll control it will be short and sweet for the majority of them. So use a water bath set to 40 degrees for all trials, it should say there. Okay, your method. The method always seems like it's going to be easy to write, and then actually when I mark, there's kind of annotations and comments all over the method because there's lots of issues. So it should be a numbered list of everything you will do in the setup and the running of your investigation, like a recipe. So it should read in exactly the same way that a recipe to make a cake would read. You will need your equipment list, so you have to include the sizes of things. So if you want a beaker, is it a 100 ml beaker, a 250 ml beaker? You need to be specific. You need to have specific volumes and concentrations of everything as well in your equipment list. It needs to be very specific and precise. Try not to miss out any steps of the process, and make sure that all of the control variables that you outlined in the previous section are also controlled within the method. So just double check that once you've written your method. Common problems with the method that I feel like I write all of the time when I'm marking is that steps are missed out, so there's just missed a section. The method might be unclear or jumps from one thing to another in the wrong order. Student tries to include too much description and justification of the steps. So in a recipe, you wouldn't read add 200 grams of sugar to make the cake sweet. So a recipe doesn't justify why we're adding things. You don't need to justify why you're doing things within the method. So just as short and sweet as possible. And don't start with number one, collect all of the equipment. Like, we know that, like, we get it. You also don't have to ever mention, and I will write this in the results table, that there is an assumption that when you take a reading, it's going to be written down, because that's what you do in a scientific investigation. So just kind of short and sweet, or clear and concise actually, rather than short and sweet. So just make it clear and concise. Don't miss any steps out, but also don't go into too much detail. And finally, risk assessment. So this used to be a slightly bigger part of the IA, but you still need a risk assessment because you have to assess the risks and the safety of what you're doing. So yeah, you need to outline safety considerations, at least 5 ideally. You can use a table like that one below. So the hazard, what could go wrong? The hazard might be glass. What could go wrong is it could fall, break, cause injuries. Who could it affect? It could affect you, the person doing the experiment. It could affect anyone else in the room if there's broken glass. How are you going to prevent it or control it? Is you are going to make sure that all of your glassware is always close to the middle of the table and not the edges of the table. And also you will have a first aid kit on hand just in case anything does happen. So it's super simple, but it needs to be in there. You should also include some ethical considerations. So if you're using human participants, think about briefs and debriefs. Think about reducing the use of plastics. How are you going to dispose of chemicals? Are you allowed to pour them down the sink? If you've done any sort of microbiology, how are you going to dispose of the plates when you're done? If you're using food stuff, is there going to be any food wastage that you could try and minimise? If you've grown any plants, then you could say that you're going to replant them afterwards so that they can grow and live a long, happy little plant life. Things like that. There's a couple of examples of risk assessment and safety there, which actually you can't read particularly well, but you get the idea.